Hello everyone, welcome back to class Biochemistry BCH 302 for the College of Natural Health Sciences Bermuda. My name is Dr. Delcina Bean Burrows and this is lecture number 2. Now for today's lecture, I should warn you ahead of time that this is going to be a very long lecture in comparison to the rest of the lectures that we will have for biochemistry. So if you need to listen to it in pieces, maybe 20-minute pieces at a time, and then maybe move on to something else and come back to it, that is perfectly fine because there is quite a bit of information that we have to get through today. So for today's lecture, at the end of this lecture, you will be able to understand what molecular biology is, you will understand what cellular biology is, and you will be familiar with the classes of biomolecules. Molecular biology is the study of biology at the molecular level. Molecular biology mainly focuses on understanding the interactions that occur between the several systems of a cell, including the interrelationship of DNA, RNA, and protein. Molecular and cellular biology are interconnected as most of the properties and functions of a cell can be described at the molecular level. So let's look at biomolecules. A biomolecule is a term that is used for molecules of more commonly ions that exist in organisms. Biomolecules include large macromolecules like proteins, carbohydrates, lipids, and nucleic acids, as well as small molecules like primary metabolites, secondary metabolites, and natural products. A more general name for this class of material is biological materials. Biomolecules are typically endogenous, but they can also be exogenous. Biomolecules contain small molecules like primary and secondary metabolites and natural products, as we stated earlier. Biomolecules consist mainly of carbon and hydrogen with nitrogen, oxygen, sulfur, and phosphorus. Among the biomolecules, nucleic acids, namely DNA and RNA, are responsible for storing an organism's genetic code, which is the sequence of nucleotides that determines the amino acid sequence of proteins, which are extremely important to life on Earth. There are 20 different amino acids that can occur within a protein. The order in which they occur plays a central role when it comes to determining the protein structure and function. Proteins themselves are key structural elements of cells. They also play the role of transporters that transport nutrients and other molecules in and out of cells, and as enzymes and catalysts for the vast majority of chemical reactions that take place in living organisms. Proteins also form antibodies and hormones, and they influence gene activity. Carbohydrates, which are also made up primarily of molecules containing atoms of carbon, hydrogen, and oxygen, are vital energy sources and structural components of all life, and they are among the most abundant biomolecules on Earth. They are built from 4 kinds of sugar units: monosaccharides, disaccharides, oligosaccharides, and polysaccharides. Another key biomolecule of living organisms are lipids. They perform a variety of roles, including serving as a source of stored energy and acting as chemical messengers. Lipids also form the membranes that separate cells from their environments and compartmentalize the cell interior, resulting into organelles like the nucleus and the mitochondrion in more complex organisms. All biomolecules have a common fundamental relationship between structure and function. This is influenced by elements like the environment in which a given biomolecule occurs. Let us now discuss the classes of biomolecules. There are four major classes of biomolecules: carbohydrates, lipids, proteins, and nucleic acids. A carbohydrate is a biological molecule or a biomolecule that is made up of carbon C, hydrogen H, and oxygen O atoms, typically with a hydrogen-oxygen atom ratio of 2:1, as seen in water. Or, better put, the empirical formula CmH2ON, where the M can be different from the N. This formula also applies to monosaccharides. Some of the exceptions that exist is the deoxyribose, a sugar component of DNA that has empirical formula C5H10O4. Carbohydrates are in principle hydrates of carbon, meaning that structurally it will be more accurate to see them as polyhydroxyaldehydes and ketones. Carbohydrates play several roles in living organisms. Polysaccharides function as the storage of energy, such as starch and glycogen, and as structural components, such as the cellulose in plants and chitin in arthropods. The 5-carbon monosaccharide ribose is an essential component of coenzymes and the backbone of the genetic molecule RNA. The associated deoxyribose is a component of DNA. Saccharides and their byproducts include many other essential biomolecules that play vital roles in the immune system, fertilization, preventing pathogenesis, blood clotting, and development. Although carbohydrates are found in a wide variety of foods, its important sources are cereals like wheat, maize, and rice, potatoes, sugar cane, fruits, table sugar, which is sucrose, bread, milk, etc. Starch and sugar are the important carbohydrates in the human diet. Starch can be found in a variety of foods in potatoes, while sugar appears in our diet largely as sucrose, which is table sugar, that is added to drinks and many prepared foods like jam, biscuits, and cakes. Glucose and fructose can be naturally found in many fruits and some vegetables. Now let's consider the structure of carbohydrates. Saccharides are in general made up of simple carbohydrates referred to as monosaccharides with the general formula CH2ON, where the N is 3 or more. A typical monosaccharide has the structure H, CHO, C equals O, CHOH, Y, H. That is an aldehyde or ketone with many hydroxyl groups added, typically one on each carbon atom that is not part of the aldehyde or ketone functional group. Though some biological substances commonly referred to as monosaccharides do not follow this formula, for example, uronic acids and deoxysugars like fructose, there are many chemicals that do follow this formula but are not thought to be monosaccharides, such as formaldehyde and N-acetyl. The open-chain form of a monosaccharide often exists alongside a closed ring form where the aldehyde ketone carbonyl group carbon, CO, and hydroxyl group, OH, react, forming a hemiacetal with a new CO-C bridge. Monosaccharides can be linked together to form polysaccharides or oligosaccharides in a large variety of ways. Several carbohydrates have one or more modified monosaccharide units that have had one or more groups replaced or removed, such as deoxyribose, which is a component of DNA, is a modified version of ribose. Carbohydrates are polyhydroxyaldehydes, ketones, alcohols, acids, their simple derivatives, and their polymers having linkages of the acetyl type. They can be categorized according to their degree of polymerization and can be divided primarily into three principal groups, namely sugars, oligosaccharides, and polysaccharides. Now let's discuss carbohydrate metabolism. Carbohydrate metabolism has to do with the various biochemical processes that are responsible for the formation, breakdown, and interconversion of carbohydrates within living organisms. The most vital carbohydrate is glucose, which is a simple sugar that is metabolized by nearly all known organisms. Glucose and other carbohydrates are a part of a broad variety of metabolic pathways across species. Plants synthesize carbohydrates from carbon dioxide and water by way of photosynthesis, storing the absorbed energy internally and most times in the form of starch or lipids. Oxidation of 1 gram of carbohydrate produces about 4 kilocalories of energy, while oxidation of 1 gram of lipids produces about 9 kilocalories. Energy obtained from metabolism is usually stored temporarily within cells in the form of ATP. Organisms that are capable of aerobic respiration metabolize glucose and oxygen to release energy with carbon dioxide and water as byproducts. Now let's take a look at carbohydrate catabolism. Catabolism is the metabolic reaction that cells go through to extract energy. There are two major metabolic pathways when it comes to monosaccharide catabolism. They are glycolysis and the citric acid cycle. In glycolysis, oligosaccharides and polysaccharides are first cut to smaller monosaccharides by enzymes called glycoside hydrolases. The smaller monosaccharide units can then enter into monosaccharide catabolism. In some cases, such as with humans, not all types of carbohydrate are usable, and they do not have the necessary digestive and metabolic enzymes. The next biomolecule that we are going to look at is lipids. Lipids are insoluble biomolecules defined by their complete lack of polarity needed for solubility in water-based solutions. Nowadays, fats are considered the same as lipids, giving lipids a negative role in diet and health. However, lipids play important roles in numerous cellular processes like storage of energy, structural support, protection, and communication. Some examples of common lipid groups are waxes, steroids, fats, and phospholipids. The fatty acid, which is a type of lipid monomer, contains one carboxyl group at the end of a linear hydrocarbon that has at least 4 carbon atoms. Since hydrocarbon chains are nonpolar, fatty acids with long hydrocarbon chains are largely insoluble in water despite having one polar functional group. Contrasting from other biomolecule groups, Fatty acid monomers are not directly bonded to each other in polymer chains. Dehydration synthesis reactions that occur in lipids form an ester linkage between the carboxyl group of a fatty acid and the hydroxyl group of an alcohol monomer like glycerol. Monomer and polymer structures vary widely based on the type of lipid. Similar to many other biomolecules, fatty acids form isomers when a double bond is present because the double bond locks the atoms around it into a fixed position. The particular isomers that are present in a specific lipid have significant effects on the lipid structure and function in living organisms. Virtually all living organisms synthesize and integrate cis fatty acids into their lipids. Cis fatty acids are isomers in which the continuing carbon chains on each end of the double bond face the same direction. A cis isomer is bent and prevents cis fatty acids from packing closely together. Trans fatty acids are isomers that are often created during commercial food production. In trans fatty acids, the continuing carbon chains face opposite directions around a double bond. Trans isomers are structurally similar to saturated fatty acids because the hydrocarbon chain does not contain a bend. Waxes are a class of lipids that have two monomers, one fatty acid bonded through an ester linkage to one alcohol, a hydrocarbon that contains a hydroxyl group. The hydrocarbon chain in the alcohol monomer of waxes diverges from a short linear chain to complex carbon ring structures. Waxes provide protective barriers that prevent water loss and protect the cells. Waxes also protect the seeds and nutrients inside plant fruits and coat the surfaces of plant leaves, creating a cuticle to prevent water loss. Waxes prevent dehydration from body surfaces of various insects and repel water on the surface of bird feathers and some animal furs. Steroids are a class of lipids that contain 4 fused carbon rings. Though steroids can bond to fatty acids, their molecules do not have a fatty acid chain, and the monomer of a steroid biomolecule can be difficult to define. Steroid rings typically have one or a few small functional groups that include hydroxyls, carbonyls, or carboxyls. Cholesterol and other steroids that contain a hydroxyl group are termed sterols. Cholesterol and other associated sterols are present in animal cell membranes and are forerunners for the synthesis of many vital steroids and other sterol derivatives. Numerous steroids and their byproducts carry out vital cellular functions. Steroid hormones like estrogen and testosterone control the reproductive processes and development. Anabolic steroids, which are a particular type of artificially manufactured steroid drugs, stimulate muscle growth and increases the development of secondary sex characteristics. In persons who suffer from metabolic diseases, anabolic steroids can improve their health by reinstating normal signals, but anabolic steroid use by an otherwise healthy individual can be very harmful to internal organ function. Now let's consider the macromolecule protein. Proteins are macromolecules that are made up of one or more long chains of amino acid residues. Proteins undertake a wide array of functions within organisms. This includes catalyzing metabolic reactions, DNA replication, responding to stimuli, and transporting molecules from one location to another. Proteins vary from one another mainly in their sequence of amino acids, which is determined by the nucleotide sequence of their genes, and which typically leads to protein folding into a specific three-dimensional structure that determines its activity. A linear chain of amino acid residues is referred to as a polypeptide. A polypeptide contains at least one long polypeptide and short polypeptides that have less than 20 to 30 residues, and they are hardly considered to be proteins and are normally called peptides or sometimes oligopeptides. Each amino acid residue is bonded together by peptide bonds and adjacent amino acid residues. The sequence of amino acid residues in a protein is defined by the sequence of a gene, which is encoded in the genetic code. Just after or even during synthesis, the residues in a protein are often chemically modified by post-translational modification, which changes the physical and chemical properties, folding, stability, activity, and ultimately the function of the proteins. Once they are formed, proteins only exist for a certain period of time and are then degraded and recycled by the cell's machinery through the procedure of protein turnover. A protein can exist for minutes or years with an average lifespan of 1 to 2 days in mammalian cells. Abnormal proteins are degraded more quickly, either due to being targeted for destruction or due to being unstable. Like other biological macromolecules, proteins are vital parts of organisms and partake in almost every process within cells. Many proteins are enzymes that catalyze biochemical reactions and are essential for metabolism. Proteins also perform structural or mechanical functions like actin and myosin in muscle and the proteins in the cytoskeleton, which form a system of scaffolding that maintains cell shape. Other proteins are central in cell signaling, immune responses, cell adhesion, and the cell cycle. Proteins are needed in animal diet because they provide the essential amino acids that cannot be synthesized. Proteins are gathered from amino acids using the information that is encoded in genes. Each protein has its own distinctive amino acid sequence that is indicated by the nucleotide sequence of the gene encoding this protein. The genetic code is a set of 3 nucleotide sets that are called codons, and each 3 nucleotide combination designates an amino acid. Because the DNA has 4 nucleotides, the total number of codons that are possible is 64. Because of this, there is some redundancy in the genetic code, with some amino acids indicated by more than 1 codon. Genes that are encoded in DNA are first transcribed into pre-messenger RNA, also known as mRNA, by proteins like RNA polymerase. Many organisms then process the pre-mRNA primary transcript using several methods of post-transcriptional modification to form the mature mRNA, which is then used as a template for protein synthesis by the ribosome. In prokaryotes, the mRNA can either be used immediately or bound by a ribosome after it has moved away from the nucleotide. On the other hand, eukaryotes create mRNA in the cell nucleus, and then translocate it across the nuclear membrane into the cytoplasm, where protein synthesis then takes place. The process of synthesizing a protein from an mRNA template is known as translation. The mRNA is loaded onto the ribosome and is read 3 nucleotides at a time by matching each codon to its base pairing anticodon located on a transfer RNA molecule, which carries the amino acid corresponding to the codon it recognizes. Let's now look at protein structure. Most proteins fold into unique three-dimensional structures, and the shape into which a protein naturally folds is known as its native conformation. Though many proteins can fold unaided, simply through the chemical properties of their amino acids, other proteins require the aid of molecular chaperones to fold them into their native states. And here we have the different aspects of protein structures. The primary structure is the amino acid sequence. The secondary structure is the often repeating local structures that are stabilized by hydrogen bonds. Because secondary structures are local, many sections of different secondary structures can be present in the same protein molecule. The tertiary structure is the general shape of a single protein molecule that is the spatial relationship of the secondary structures to one another. Tertiary structure of protein is generally stabilized by non-local interactions, most commonly through the formation of a hydrophobic core, but also through salt bridges, hydrogen bonds, disulfide bonds, and even post-translational modifications. The tertiary structure is what controls the basic function of the protein. The quaternary structure is the structure formed by several protein molecules, also known as polypeptide chains, typically referred to as protein subunits in this context, which function as a single protein complex. In addition to these different structures, proteins can move between several related structures while they perform their functions. In this context, these tertiary and quaternary structures are usually called conformations, and transitions between them are called conformational changes. Now let's take a look at enzymes. The most common function of proteins in the cell is as enzymes that catalyze chemical reactions. Enzymes are typically highly specific and accelerate one or a few chemical reactions. Enzymes undertake almost all the reactions that are involved in metabolism, as well as manipulating DNA in procedures like DNA replication, DNA repair, and transcription. Certain enzymes act on other proteins to add or remove chemical groups in a process called post-translational modification. Around 4,000 reactions are known to be catalyzed by enzymes. The molecules bound and acted upon by enzymes are termed substrates. Though enzymes may contain hundreds of amino acids, it is usually only a small portion of the residues that come in contact with the substrate, and even a smaller portion, an average of 3 to 4 residues, that are directly involved in catalysis. The region of the enzyme that binds the substrate and has the catalytic residue is called the active site. Let's now consider cell signaling and ligand binding. Proteins like insulin are extracellular proteins that transmit a signal from the cell in which they were synthesized to other cells in distant tissues. The others are membrane proteins that play the role of receptors whose key function is to bind a signaling molecule and induce a biochemical response in the cell. Numerous receptors have a binding site exposed on the cell surface and an effector domain within the cell. Antibodies are protein components of an adaptive immune response. Their key function is to bind antigens and foreign substances in the body and target them for destruction. Antibodies can be secreted in the extracellular environment or anchored in the membranes of specialized B cells called plasma cells. Ligand transport proteins bind certain small biomolecules and transport them to other locations in the body of a multicellular organism. These proteins usually have a high binding affinity when their ligand is present in high concentrations, but must also release the ligand when it is present at low concentrations in the target tissues. A recognized example of ligand binding protein is the hemoglobin. The hemoglobin transports oxygen from the lungs to other organs and tissues in all vertebrates and has close homologs in all biological kingdoms. Transmembrane proteins may also serve as ligand transport proteins that change the permeability of the cell membrane to small molecules and ions, as the membrane in itself has a hydrophobic core through which polar or charged molecules cannot diffuse. Structural proteins give stiffness and rigidity to biological components that are otherwise fluid. For example, collagen and elastin are essential components of connective tissues like cartilage and keratin that are found in hard or filamentous structures like the hair, nails, feathers, hooves, and some animal shells. A number of globular proteins can also play structural functions. For example, the actin and tubulin are globular and soluble as monomers, but polymerize to form long, stiff fibers that make up the cytoskeleton, which allows the cell to uphold its shape and size. Other proteins that play structural roles are motor proteins like myosin, kinesin, and dynein, which are able to generate mechanical forces. These proteins are essential for cellular motility of single-celled organisms and the sperm of many multicellular organisms that reproduce sexually. They also produce the forces exerted by contracting muscles and play vital roles in intracellular transport. So the next biomolecule that we will consider is nucleic acids. Nucleic acids are molecules that allow organisms to transfer genetic information from one generation to the next. There are two types of nucleic acids: deoxyribonucleic acid, which is DNA, and ribonucleic acid, which is RNA. Nucleic acids are biopolymers that are vital to all known forms of life. They are made up of monomers, which are nucleotides made of three components: a 5-carbon sugar, a phosphate group, and a nitrogenous group. When the sugar is simple ribose, the polymer is RNA, and when the sugar is derived from ribose, such as deoxyribose, the polymer is DNA. Nucleic acids are the most vital of all biomolecules and are found in all living things where they create and code and then store information in the nucleus of living cells. They also transmit and express the information that is inside and outside the cell nucleus to the internal operations of the cell and eventually to the next generation of each living organism. The encoded information is enclosed in and carried through the nucleic acid sequence. This provides the structure of nucleotides within the molecules of RNA and DNA. Strings of nucleotides are bonded to create helical backbones, usually one for RNA and two for DNA, and organized into chains of base pairs chosen from the 5 primary or canonical nucleobases: cytosine, guanine, adenine, thymine, and uracil, with the thymine only occurring in DNA and the uracil only occurring in RNA. By means of amino acids and protein synthesis, the precise sequencing in DNA of these nucleobases pairs allows the storing and transmitting of coded instructions as genes. In RNA, base pair sequencing allows the creation of new proteins that determine the frames, parts, and most chemical processes of all life forms. The DNA is a polymer of the 4 nucleotides A, C, G, and T that are joined via a backbone of alternating phosphate and deoxyribose sugar residues. These nitrogen-containing bases happen in complementary pairs as determined by their capability to form hydrogen bonds between them. A always pairs with T via 2 hydrogen bonds, and G always pairs with C via 3 hydrogen bonds. The spans of A to T and G to C hydrogen bonded pairs are almost identical, allowing them to link the sugar-phosphate chains uniformly. These structure plus the molecule's chemical stability makes the DNA the idyllic genetic material. The bonding amid the complementary bases also offers a mechanism for the replication of DNA and the transmission of genetic information. The RNA is a single-stranded nucleic acid polymer of the 4 nucleotides A, C, G, and U, combined through a backbone of alternating phosphate and ribose sugar residues. It is the first intermediary when transforming the information from DNA into proteins vital for cell function. Some RNAs also play direct roles in cellular metabolism. RNA is created by copying the base sequence of a segment of double-stranded DNA, referred to as a gene, into a piece of single-stranded nucleic acid. This process, referred to as transcription, is catalyzed by an enzyme called RNA polymerase. Let's now consider the different types of RNA. The messenger RNA, or mRNA, delivers encoded information in one or more genes from the DNA to the ribosome, a specialized structure or organelle where that information is decoded into a protein. In prokaryotes, mRNAs have a precise prescribed copy of the original DNA sequence with a terminal 5, triphosphate group, and a 3-hydroxyl residue. In eukaryotes, the mRNA molecules are more intricate. The 5-triphosphate residue is further esterified, creating a structure termed a cap. At the three ends, eukaryotic mRNAs usually have a long runs of adenosine residues, which is poly A, that are not encoded in the DNA, but are added enzymatically after transcription. Eukaryotic mRNA molecules are typically made up of small sections of the original gene and are created by a process of cleavage and rejoining from an original precursor RNA, which is the pre-mRNA molecule, which is a precise copy of the gene. Overall, prokaryotic mRNAs are degraded very quickly, while the cap structure and the poly-A tail of eukaryotic mRNAs significantly increase their stability. Now let's consider ribosomal RNA, or rRNA. rRNA molecules are the structural components of the ribosome and form wide secondary structures that play active roles in recognizing preserved portions of mRNAs and tRNAs. The rRNA also help with the catalysis of protein synthesis. In the prokaryote E. coli, 7 copies of the rRNA gene synthesize around 15,000 ribosomes per cell. The numbers are much larger in eukaryotes, somewhere between 50 to 5,000 sets of rRNA genes, and as much as 10 million ribosomes can be present in a single cell. In eukaryotes, these rRNA genes are coiled out of the main chromosomal fibers and combined in the presence of proteins to create an organelle termed the nucleolus. The nucleolus is where the rRNA genes are transcribed and the initial assembly of ribosomes take place. Now let's consider tRNA, or transfer RNA. tRNA convey individual amino acids into the ribosome for assembly into the mounting polypeptide chain. The tRNA molecules have 70 to 80 nucleotides and fold into a distinguishing clover leaf structure. Specific tRNAs exist for each of the 20 amino acids needed for protein synthesis, and in most cases, more than one tRNA for each amino acid is present. The nucleotide sequence is changed into a protein sequence by translating each 3-base sequence, referred to as a codon, with a precise protein. The 61 codons used to code amino acids can be read by no fewer than 61 individual tRNAs. Thomas Sachs and Sidney Altman discovered that certain RNAs, now known as ribozymes, showed enzymatic activity. Sachs demonstrated that a non-coding sequence, which is an intron, in the small subunit of rRNA of proteasomids, which had to be removed from the rRNA, was functional, can remove itself from a much longer precursor RNA molecule and rejoin the two ends in an autocatalytic reaction. Altman demonstrated that the RNA section of an RNA protein complex, referred to as a ribonuclease P, can splice a precursor tRNA to create a mature tRNA. Aside from self-splicing RNAs akin to the one discovered by Sachs, artificial RNAs have been created that show a number of catalytic reactions. It is now widely accepted that at a stage during evolution, the RNA catalyzed and stored genetic information. Now let's take a look at antisense RNAs. Almost all antisense RNAs are synthetically modified byproducts of RNA or DNA with possible therapeutic value. Antisense RNAs comprise of sequences that are counterpart of the normal coding sequences found in mRNAs. Similar to mRNAs, antisense RNAs are single-stranded, but they cannot be translated into protein. Antisense RNAs can deactivate their complementary mRNA by creating a double-stranded structure that blocks the translation of the base sequence. Artificially introducing antisense RNAs into cells can selectively inactivate genes by interfering with normal RNA metabolism. Let us now discuss viral genomes. Many viruses use RNA as their genetic material. This is predominant amongst eukaryotic viruses. Some popular examples are human immune deficiency virus, HIV, poliovirus, and influenza virus in humans, and tobacco mosaic virus in plants. In some viruses, the entire genetic material is encoded in a single RNA molecule, while in the segmented RNA viruses, several RNA molecules may be present. Many RNA viruses like HIV use a particular enzyme called reverse transcriptase that allows the replication of the virus through a DNA intermediate. At times, this DNA intermediate can become incorporated into the host chromosome during infection. The virus then exists in a latent state and can effectively evade the host immune system. So we will now, at this stage of the lecture, discuss techniques of molecular biology, and specifically we're going to talk about the polymerase chain reaction, or the PCR. The polymerase chain reaction is a technique that is used for copying DNA. The PCR technique allows a particular DNA sequence to be copied or modified in prearranged ways. The reaction is very powerful, and it could even magnify one DNA molecule into 1.7 billion molecules in less than 2 hours. This technique can be used to introduce restriction enzyme sites to ends of DNA molecules or to mutate specific bases of DNA. This is a method that's called site-directed mutagenesis. The PCR technique has many variations, like the reverse transcription PCR used for the amplification of RNA, and quantitative PCR, which is used for quantitative measurement of DNA or RNA molecules. Gel electrophoresis is another molecular biology technique. Gel electrophoresis is a principal tool when it comes to molecular biology. The rudimentary principle here is that DNA, RNA, and proteins can all be separated using an electric field and size. Under agarose gel, electrophoresis DNA and RNA can be separated based on size by running the DNA through an electrically charged agarose gel. Proteins can be separated based on size by using an SDS page gel, or based on both size and electric charge by using a 2D gel electrophoresis. Microarrays is another molecular biological technique. DNA microarray is a pool of spots that is attached to a solid support like microscope slides, where each spot has one or more single-stranded DNA oligonucleotide fragments. Arrays make it possible to put down large amounts of very small spots on a single slide. Each spot contains a DNA fragment molecule that is paired to a single DNA sequence. A deviation on this technique allows the gene expression of an organism at a specific stage in development to be qualified. This is called expression profiling. Since several arrays can be created with precisely the same position of fragments, they are very useful for comparing the gene expression of two different tissues, like a healthy and cancerous tissue. Arrays can also be made with molecules other than DNA. The next technique that we will consider is allele-specific oligonucleotide. This is a technique that allows the detection of single base mutations without the use of PCR or gel electrophoresis. Short and labeled probes are exposed to non-fragmented target DNA. Hybridization takes place with high specificity because of the short length of the probes. The target DNA is then washed and the probes that did not hybridize are removed. After this, the target DNA is then examined for the presence of the probe through radioactivity or fluorescence. In this experiment, control must be exercised to guarantee successful experimentation. We will now move on to a discussion in cellular biology. Cell biology is the study of the structure of cells and its function. It is based on the concept that the cell is the fundamental unit of life. Concentrating on the cell allows for a thorough understanding of the tissues and organisms that cells contain. Some organisms have just one cell, whereas others are divided into cooperative groups with vast numbers of cells. Generally speaking, cell biology focuses on the structure and function of a cell, from the universal properties shared by all cells to the exclusive and highly intricate functions specific to particular cells. The study of cells is carried out at a molecular level, though most of the processes in the cell are made up of a mix of small organic molecules, inorganic ions, hormones, and water. Around 75 to 85% of the cell's volume is water, making it a crucial solvent as a result of its polarity and structure. These molecules within the cell create a suitable environment for the cell to undertake metabolic reactions and signaling. The cell shape differs amongst the various types of organisms and are divided into two categories, eukaryotes and prokaryotes. Eukaryotic cells are made up of animal, plant, fungi, and protozoa cells, and the shapes are usually round and spherical, while prokaryotic cells are made up of bacteria and archaea. The shapes are spherical, such as the cocky, the rods, bacillus, the curved, vibrio, and the spirals, spiral sheets. Cell biology lays more emphasis on the study of eukaryotic cells and their signaling pathways, while prokaryotes are covered under microbiology. The key elements of the general molecular composition of the cell are proteins and lipids, which are either free-flowing or membrane-bound, along with other different internal sections called organelles. The cell environment is made up of hydrophilic and hydrophobic regions that allow for the exchange of molecules and ions. The hydrophilic sections of the cell are mostly on the inside and the outside of the cell, while the hydrophobic sections are within the phospholipid bilayer of the cell membrane. The cell membrane is made up of lipids and proteins, which explains its hydrophobicity because it is a nonpolar substance. Consequently, for these molecules to partake in reactions within the cell, they need to be able to cross this membrane layer to enter into the cell. Access to the cell is gained via osmotic pressure, diffusion, concentration gradients, and membrane channels. So when we study cellular biology, we have to ask ourselves, what is a cell? Well, a cell is the basic structural, functional, and biological unit of all known living organisms. A cell is the smallest unit of life that can reproduce individually. Cells are made up of cytoplasm that are enclosed within a membrane that contains numerous biomolecules like proteins and nucleic acids. Organisms can be classified as either unicellular, having a single cell, or multicellular, such as in plants and animals. While the number of plant and animal cells vary from species to species, humans have more than 10 trillion cells. Most animal and plant cells are only visible under a microscope with dimensions between 1 and 100 micrometers. Individually, the cell can metabolize its own nutrients, synthesize several types of molecules, provide its own energy, and replicate itself in order to produce successive generations. The cell can be regarded as an enclosed vessel within which countless chemical reactions take place concurrently. In a multicellular organism, cells are specialized and perform different functions through the process of differentiation. To do this, each cell is constantly communicating with its neighbors. As it collects nutrients from and ejects waste into its environs, it follows and cooperates with other cells. There are two types of cells: eukaryotic cells, which contain a nucleus, and prokaryotic cells, which do not. Prokaryotes are single-cell organisms, while eukaryotes can either be single-cell or multicellular. The table you see illustrated here gives an approximation of the chemical composition of a typical mammalian cell. Aside from water, which makes up 70% of a cell's mass, a cell is made up mostly of macromolecules. By far, the largest portion of macromolecules are proteins. Let's take a closer look at the cell membrane. A thin membrane that usually between 4 to 10 nanometers in thickness surrounds every living cell, demarcating the cell from the environment around it. Walled by its cell membrane are the cell's components, which are often large, water-soluble, highly charged molecules like proteins, nucleic acids, carbohydrates, and substances that are involved in cellular metabolism. On the outside of the cell's water-based environment are ions, acids, and alkalis that are toxic to the cell, as well as nutrients that the cell needs to absorb in order to live and grow. Therefore, the cell membrane has two functions. The first one is to be a barrier that keeps the components of the cell and toxic substances out. The second is to be the entrance that allows passage into the cell of vital nutrients and removal of waste products from the cell. Now let's look at the chemical composition and membrane structure. The study of cells has shown that all membranes are made up of proteins and fatty acid-based lipids. Membranes that are directly involved in metabolism have a higher percentage of protein. Membrane lipids are two main phospholipids and sterols. They dissolve readily in organic solvents, but they both have a section that is attracted to and soluble in water. This amphiphilitic property is rudimentary to the function of lipids as building blocks of cellular membranes. There are two types of membrane proteins. The first one, called the extrinsic proteins, is loosely attached to ionic bonds or calcium bridges to the electrically charged phosphoryl surface of the biolayer. They can also attach to the second type of protein, referred to as the intrinsic proteins. These intrinsic proteins are tightly embedded within the phospholipid biolayer. Transport across the membrane. The chemical structure of the cell membrane makes it very flexible, but the membrane is also a tough wall that allows some dissolved substances to pass while blocking the others. Transport of essential substances is carried out by a specific class of intrinsic proteins that form a range of transport systems. Particles that are too big to be diffused or pumped are usually swallowed or disgorged whole by an opening and closing of the membrane. The principle of diffusion is responsible for this movement of solutes across the cell membrane. According to the principle of diffusion, a dissolved substance diffuses down a concentration gradient that is without influence from outside source. It moves from a point where its concentration is high to a point where its concentration is low. Diffusion continues down this gradient until a state of equilibrium is reached. At this point, there is an equal concentration of both points and an equal random diffusion in both directions. During diffusion, the solute loses free energy so that by the time it reaches equilibrium at a lower concentration, it is unable to return spontaneously to its former high concentration. Permeation. Permeation is the diffusion through a barrier of a substance in solution. Permeability is measured as the rate at which a particular substance in solution crosses the membrane. The permeability of a cell membrane increases in parallel with the permeant's ability to dissolve in organic solvents. This has led researchers to conclude that permeability is a function of fatty acids interior of the bilipid layer rather than its phosphoryl exterior. This ability to dissolve in organic solvents rather than in water is given a unit of measure that is called the partition coefficient. The higher the solubility of a substance, the higher its partition coefficient, and the higher the partition coefficient, the higher the permeability of the membrane to that specific substance. The presence of internal membranes is what differentiates eukaryotic cells from prokaryotic cells. Prokaryotic cells are small and contain only a single cell membrane, while eukaryotic cells are much larger, with the cell membrane making up only 10% or less of the total cellular membrane. Now let's look at cellular organelles and their membranes. The centrosome is an associated pair of cylinder-shaped protein structures called centrioles that organize microtubules and help in forming the mitotic spindle during cell division in eukaryotes. Cell membrane or plasma membrane is the part of the cell that separates the cells from the outside environment and protects the cell whilst regulating what goes in and out of the cell. The cell wall is an extra layer of protection and it gives structural support. This is only found in plant cells. The chloroplast is the key organelle for photosynthesis, and this is only found in plant cells. Cilium is a motile structure of eukaryotes that have cytoskeleton. Cytoplasm refers to the contents of the main fluid-filled space inside the cells and is a jelly-like substance where chemical reactions also happen. Cytoskeleton are the protein filaments inside cells, and these are microfilaments, microtubules, and intermediate filaments. The endoplasmic reticulum, or ROF, is the major location of membrane protein synthesis. Endoplasmic reticulum, which is smooth, or smooth is the major location of lipid synthesis. Endosomes are the vesicles that traffic membrane and intra and extracellular substances for recycling and degradation by lysosomes. Flagellum is a motile structure of bacteria, archaea, and eukaryotes. The Golgi apparatus is the site of protein glycosylation in the endomembrane system. The bilipid layer is the fundamental organizational structure of cell membranes. The lysosome is the acidic organelle that breaks down cellular waste products and debris into simple compounds, and this is only found in animal cells. The microvilli increases the surface area for absorption of nutrients from surrounding environments. The mitochondrion is the major energy-producing organelle that releases energy in the form of ATP. The nucleus contains chromosomes that are composed of DNA. The organelle is a term used for the main subcellular structures. Peroxisomes is a small organelle that uses oxygen to break down and detoxify long fatty acids and other molecules. Pili, also known as fimbria, is used for conjugation and sometimes movement. The ribosome is the RNA and protein complex that is required for protein synthesis in cells. The starch grain is found in the cytoplasm of a typical plant cell, and it stores chemical energy of the plant. The vacuole comprises of the cell sap or other storage material. And the vesicle are small membrane-bound spheres inside the cells that transport substances. Okay, so we've reached the end of our lecture finally. So for assignment number 2, in an essay of no less than 250 words, you're going to provide descriptions for molecular biology and cellular biology. What are the techniques of molecular biology, and how might these techniques, in your opinion, shape or influence natural health practice? As always, your assignments are due no later than 11:55 p.m. on this coming Sunday. And if you have any difficulty at all with this assignment, please shoot me an email or send me a text message to 703-7327. And other than that, do have a good week.